Okay, our number of participants seems to have stabilized, so let's get underway. So Happy New Year. I feel that I can say that until probably the end of this week, and then it's starting to get a little bit old by after like the 11th of January. But Happy New Year. I hope everyone had a wonderful holiday break. So welcome to the first BioCurator work group call for 2025. We concluded our 2024 series by having a presentation to celebrate all of our volunteers. And I want to start 2025 by again thanking you all for your continued commitment to what really is an effort that's driven by the contributions of our volunteers. So thank you for signing on again for another year and for being here today. A quick note before we get into today's presentation, on the first meeting in February, so the February 12th meeting, we're going to be having another one of our Q&A style sessions with the topic of gene curation. And so we sent out a request, I think last week, and we'll send it out again, and the links there, we'll put it in the chat, but asking you to submit any questions that you might have around gene curation and absolutely anything you can think of to do with gene curation. So burning questions, random thoughts you might have, something that you've thought about but not been sure of and not sure of the right forum to ask it in, this is the time. That session really will be driven by the questions we receive. So submit them, anything you can think of. And we're asking for all of those submissions to be in by January 24. Like I said, we'll put the link to that suggestion form in the chat as well. But if no one else has general announcements that are relevant for this forum, I will move on to our presentation for today. So today we're going to be joined by Melissa Landrum, who will be giving us sort of an introduction to, although I'm sure people are very familiar already with the ClinVar database, and some updates for recent features of ClinVar. So with that, I'll hand over to Melissa.
All right, thank you, Alicia. Let me share my screen. And do you see PowerPoint?
Yes, it's in the editing mode at the moment, not presenter view, but
yeah. And how about now?
Now it is. Perfect.
Great. Okay, thank you. So today I'm going to talk about navigating the ClinVar database. And I'm going to do this in three major sections. One is understanding the types of data that you see in ClinVar as you navigate the database. Second will be actually searching for data that you care about in ClinVar. And then finally, we'll navigate our way through the ClinVar variant page. So let's start by understanding the types of data that you see in ClinVar. And let's just give a little bit of background here before we really get into it. So many of you are familiar with ClinVar, but it is the NIH database of classifications of genetic variants relative to genetic disease, cancer, and drug responses. The database today has more than 4.7 million submitted records for more than 3 million variants that have come to the database from more than 2,900 submitters across the world. Now, you may think of ClinVar as a ClinGen product. ClinVar and ClinGen work closely together, but it's actually not a ClinGen product. It's a product of NCBI, which is part of the National Library of Medicine at the NIH. NCBI is the group that provides databases like PubMed, the Genetic Testing Registry, clinicaltrials.gov, and dbSNP. So in ClinVar, the data that you see are considered variant-level classifications as opposed to patient-level classifications. We'll talk a little bit more about that. It's a submission-driven database, meaning that if a variant in a classification is going to be in ClinVar, it must be submitted to us. We don't have staff who are classifying variants or pouring the literature for this information. It must be submitted to the database. And then finally, all of the data that you see in ClinVar is fully public and available for anyone to use. So what kind of data do you see in ClinVar for variants? So we have a pretty broad scope for the kinds of variants that you see in ClinVar. It's all the way from single nucleotide variants and short indels through exon or gene deletions or duplications, up through large structural variants like copy number variants or translocations. These variants must be localized on the genome. And almost always the variant that you see, a variant that you see classified in ClinVar is a single variant. So that's as opposed to a haplotype or a genotype. We do support those. Occasionally, it's necessary to classify, two or more variants together in combination, but almost always the classifications that you see in ClinVar are for a single variant. So there are a few kinds of variants that are not in scope for ClinVar. We don't take variants that are described only as the protein change. It must be described as the nucleotide change. We don't take variants that are described only by cytogenetic bands. So like we wouldn't expect something that's just like a 20q deletion. And we don't take variants with legacy nomenclature that we can't map to a current genome assembly. So in the past, we have accepted all of these. So you may see some older records that look like these cases, but we don't accept these anymore. So every variant in ClinVar is classified for some kind of condition. And so the kinds of conditions that you might see on a classification include inherited diseases, cancer, and drug responses. So typically a variant is classified for just a single condition, a single disease or single drug response. But sometimes you will see a variant that's classified for multiple conditions, and that's usually one of these three cases. So I think the most common one is that the variant is in a gene that's associated with several diseases, but the submitter doesn't really know which disease is the right one to associate with the variant. So let's say it's a clinical testing lab that tests a gene because they know it's associated with several diseases. They have a variant in that gene that they think is pathogenic, but they don't know the patient's phenotype. They don't really know which disease that variant is pathogenic for. So when they submit to ClinVar, they may classify that variant for the whole set of diseases that gene is associated with, and there's a tag on it that might say there's some uncertainty. I don't know which of these is correct. A very infrequent, possibly never occurring case is that a variant may cause two co-occurring diseases. We think this is extremely rare. We think it may be valid for some copy number variants. So let's say you have a CNV, a large copy number loss that spans several genes, and let's say two of those genes are associated with haploinsufficiency. So if both genes are deleted, maybe that patient has two diagnoses. Theoretically, that could occur. I don't know if it actually has occurred in ClinVar. I have yet to hear of an example where a small variant like an SNV causes two co-occurring diseases, but theoretically, that could happen. And then finally, this case happens not so infrequently. So occasionally there's a variant that causes a new disease that's not yet named, but it's defined by a set of clinical features. So when that data gets submitted to ClinVar, the variant is submitted with multiple conditions, multiple clinical features. So that might look like multiple HPOIDs. So essentially, those features just define a single disease, but we don't have a single name for them. So it might look like they've classified the variant for several conditions, but it really represents a novel disease. So there are a few things that are not in scope for conditions that a variant in ClinVar would be classified for. We don't take infectious diseases, so genetic variants do not cause infectious disease. You might see susceptibility to an infectious disease. So you might see variants that cause susceptibility to, say, malaria or COVID-19. And we don't expect variants to be classified for patient phenotypes. So I mentioned this earlier, variants in ClinVar should be have a variant level classification. So a classification for the condition across all patients that you've seen the variant in. It should not be an interpretation that's specific to a patient and their phenotypes. That said, it's actually very difficult for us to distinguish between a submission that has given us the patient's phenotypes for the classification and a submitter who's trying to define a new disease. So we have a lot of conversations with our submitters trying to distinguish these two cases. And in the past, we have certainly had some people submit patient phenotypes in error, but this is something we are working on correcting. So the main thing that you see in ClinVar is the classification of the variant. And there's essentially three types of classifications that we have in ClinVar with a little bit more complication. So the first of these is pathogenicity for germline variants, and these are typically for inherited diseases. Then we have two types of classifications that come in for somatic variants. These are clinical impact and oncogenicity. These classifications would be made for cancer. And then the complication is that in the germline field, so along with classifications of pathogenicity, you might see other types of classifications that are not the ACMG terms for pathogenicity. You might see other things like drug responses. You might see classifications of variants for diseases of somatic mosaicism. You might see some other kinds of classifications like protective or association. These are not that common, but you might see them sometimes in this germline field. So I want to make sure you're aware that those are also there. Okay, so I want to talk a little bit about a couple of expectations that we have for the data and some exceptions. So variants in ClinVar are expected to have a classification for some disease. So we're expecting to only have classified variants. But there are some important exceptions, and that's when a variant is submitted to ClinVar because it has a type of evidence that is critical for classification. So the submitter doesn't have a classification, but they have a really important piece of evidence they want to make available to you as you're classifying variants. So this could be submitting a variant so that there's a citation that mentions it in ClinVar. It could be submitting a variant only with functional data, so no classification for disease, but just some relevant functional data, or it could be providing phenotypic data for individuals with that variant. So I do want to clarify that we're not expecting to get submissions of variants that were observed in a patient and just not classified. That is just not in scope for ClinVar. We're really expecting variants that have been classified for disease, or they specifically have one of these types of evidence that's important for classification. So citation, functional data, or phenotypic data. So another expectation is that variants in ClinVar are classified for a condition. So for example, the ACMG guidelines specify that when you classify a variant, you should say what disease you're classifying it for. And again, in ClinVar, we do have some exceptions. The first of these is that sometimes a variant is generally classified as uncertain or benign. So let's say you're looking at a variant and the allele frequency is so high that you think it's just unlikely that it's going to cause any disease. And so the submitter might consider it to be generally benign, and they don't even want to specify that it's benign for a particular disease. They think it's generally benign. In that case, instead of giving us a disease name, they would literally give us the words not specified as the condition. A second case is a variant may be classified for a specific condition, but the laboratory can't provide that piece of information to ClinVar. So this is usually just something, a logistic issue that maybe they store the disease name on the patient information, not the variant information, and it's difficult for them to pull the pieces together. But in this case, usually there is a condition, it's just not provided to ClinVar. And so instead of giving us a disease, they give us the words not provided as the condition. Sometimes if you contact the laboratory, they can actually tell you what the condition is. And then the third case is less common, and you'll see this more often on copy number variants. So particularly for non-recurrent copy number variants, the condition that's associated with it is patient specific. The variant is specific to the patient. And so they have a phenotype that is patient specific, and there's no overarching disease name that the laboratory can associate with it. So one option is just to say that there's no disease that name there. It's not provided. But sometimes the laboratory actually has phenotype data. They have observed clinical features that they can provide. And so they don't want to tell the user that there's nothing provided. They actually want to tell the user, please go look at the case level data to find the clinical features for this patient. So sometimes in this case, the condition name might be C cases, which is an instruction to tell you to please look at the case level data. Okay, let's talk a little bit about who submits all of these data to ClinVar. So in broad strokes, these are the kinds of groups who submit data to ClinVar. The vast majority of the data in ClinVar are submitted by clinical genetics testing laboratories. We also get data from research laboratories. This might be research labs who are doing clinical research and they're actually classifying variants. It could also be laboratories who are generating functional data. We get some data from clinical providers. This is usually one of two things. It could be that the clinician is providing phenotype data for one of their patients who's had genetic testing. Or it could be that the provider is providing their own classification of a variant, and they think they can do maybe a slightly better job than the laboratory because they have phenotype information not available to the test. We also get some data from patient registries. This is primarily data that comes from Genome Connect, which is ClinGen's patient registry, and other registries that they work with. And these submissions are also primarily focused on providing phenotype data that comes from the patients. We get data from other databases like OMIM, Gene Reviews, some of the LOVID databases, and Civic is now providing data for somatic variants. Of course, you all know we get data from the expert panels that are maintained by ClinGen, and we have a small number of variants that come from practice guidelines. If you're ever interested in knowing specifically who submits to ClinVar, we have our submitter list on the website. The URL is shown here, but there's also a link to it directly from our homepage. So the list shows you every submitter to ClinVar and some stats for each of them. And if you click on their name, you go to their submitter page, which gives you some general information about that submitter, summary information about what they've submitted, and then there's a link out to see all of their data. Okay, let's talk generally about what data is in a ClinVar submission. So this little infographic gives you a summary. One thing I'll note is that each submitted record in ClinVar gets an accession number beginning with SCV. So I'm going to be referring to submitted records as SCV throughout this talk. So each submitted record or SCV comes from a submitter. That's part of the definition of the record. And that submitter tells us the variant that they classified, what condition they classified it for, and what is that classification. There's also a review status associated with that classification that ClinVar calculates, and we'll talk a little bit more about that in a later slide. So there's a little more information that the submitter might give us about that classification. So they might tell us, give us their assertion criteria. So this is the rules of classification that they use. They might tell us the date they last evaluated the classification. They might provide a comment on the classification. This is the blurb that you see in ClinVar that explains the rationale for that classification. And they might provide citations that are relevant, like PubMed IDs that they consulted. Based on when the submissions came to us, each SCV has a date when this classification first appeared in ClinVar and the date that it was last updated. And then every submitted record in ClinVar also has some information about how the submitter observed that variant. So we're not expecting hypothetical variants in ClinVar. We're expecting to see variants that the submitter observed in some way, and that's what they based their classification on. So every submitted record in ClinVar has at least one observation, but it might be multiple, and they can vary in how data rich they are. So this part of the graphic gives you a few examples for what observations might look like. The thing you'll see most often from clinical testing laboratories is this example that we've called the germline observation from a germline variant submitted by a clinical testing lab. The clinical testing labs typically give us very little information about the observation. They basically just tell us they collected the data through clinical testing. The origin of the variant was that it's from the germline as opposed to somatic tissue. And the affected status of individuals with this variant is unknown. So that's a very typical observation from a clinical testing laboratory. In contrast, a research lab may have consent to provide a lot more information, a lot more case level information. So this example on the right represents, let's say, a variant submitted by a research laboratory. They're looking at a trio, a variant identified in a trio, and they can actually give us 3 observations, one that's specific to the proband, one specific for each parent. And they could give us those minimal fields as well as telling us something about zygosity, the sex, and maybe even the clinical features in each person. A submission that's focused on functional data might have other kinds of information in the observation. So in that case, it might be more focused on telling us the method that they use to generate their data, a summary of their result, and a citation for more information. And this last example is a somatic variant submitted by a clinical testing lab. And this could look very similar to the germline variant from a testing lab. So it could be very minimal, just indicating that the origin is from somatic tissue. There's a couple other fields that we have that are specific to somatic variants. So whether that somatic variant was present in normal tissue and the somatic variant allele fraction. So the take-home message here is that every submitted record, every SCV in ClinVar has at least one observation describing how the submitter observed that variant, and it may have more observations, more than one. It could be one or many, and each of those observations may be minimal or they may be very data rich. So all of these submitters are providing all of this data to ClinVar, and then what does ClinVar do with that submitted data? So the first thing we do is we validate data. So we validate individual pieces of data in the submission as well as combinations of data. And we also enforce data standards. So we do things like check to make sure that your HTTPS expression is valid. The next step is we aggregate data. So we aggregate submissions for the same variant into a VCV record. We aggregate submissions for the same variant condition pair into an RCV record. And we calculate a review status for each record, for the submitted record, the SCV, and for each of the aggregate records, the RCV and the VCV. All of those get a review status. And then we also calculate an aggregate classification for the RCV and VCV record. So we'll talk more about all of this. We also archive submitted data. So as I mentioned earlier, every submitted record to ClinVar gets assigned an SCV accession number. And that accession number also gets a version number, which means we can track changes to that submitted record. So let's say you submit your classification of a variant today. In 6 months, you have more evidence, so you update the record to add that new evidence. And then maybe next year, you reclassify the variant and you update it then. In that case, your version number would change every time so that we can tell when you first submitted it, here's what your record looked like. When you updated it each time, here's what that update looked like. And then finally, of course, we publish and make the data available. We release data every week and every month, and our monthly releases are the ones that we archive indefinitely. So the step here that's the most complicated is this one about aggregation and calculation. So I'm going to talk a little bit more about these steps. So first of all, as I mentioned, we calculate a review status for each submitted record, so for each SCV. And let me tell you why this is important. So as you probably know, submissions to ClinVar are of varying quality. And it's important both to us for later steps, which I'll describe to you, and to our users, that you have a way to distinguish between the varying levels of quality of records in ClinVar. Now, we know from talking to our users that each of you has your own way of evaluating the quality. Often that's a very manual process and it's quite subjective. For our purposes, we need to have a method that is automated and that is objective. And so the way that we've done this is we developed this field called the review status, and we calculate this review status to give you an idea of how much review went into the classification on this record. That's kind of a proxy for how trustworthy is it. So the way we calculate this is we look at the SCV and we ask, is it from a practice guideline? If so, that gets the highest review status, which is called practice guideline, and that's considered a 4-star record. There's very few of those. So the next question we ask is the SCV from an expert panel? If it is, that submitted record is considered a 3-star record, and the review status is reviewed by expert panel. So I think we have about 18,000 variants that are expert panel reviewed today. So that's great, but it's also not the vast majority of data in ClinVar. So typically we go to the next question, which is, does the SCV have assertion criteria and evidence for that specific classification? As an alternative, the submitter can provide their contact information, which is a way of telling the user that they can contact them to get the evidence. But really what we're getting at here is transparency. Did the submitter provide transparency into their classification rules and the specific evidence for this classification? If the answer is yes, that's a better, more trustworthy record. And so that is considered a 1-star submission, and the review status is called criteria provided single submitter. So this is shorthand for they provided their assertion criteria and their evidence, and it's from a single submitter as opposed to a panel of experts. If this submitted record did not provide assertion criteria or their evidence, then it's considered a 0-star record, and the review status is typically no assertion criteria provided. There are the few cases that I mentioned earlier. There are some submissions where we don't get a classification at all for functional data or literature. And in that case, the review status is no classification provided. So you can see this is how we calculate more trustworthy to potentially less trustworthy records. But I will note, just because it's 0 star doesn't mean it's a bad record. It just means based on these objective criteria, we don't have as much transparency into the classification that they provided. Okay, so we calculate a review status for the submission. We've done all of our validation on the data. The next step for us is to aggregate the data. So this infographic walks you through that. So each of the gray boxes here represents a submitted record. So you see we have 4 SCVs, SCV 1, 2, 3, and 4, and they come from 4 different submitters. But you can see they're all for the same variant. So they're all for variant 1. So when we aggregate data by variant, we would aggregate all 4 of these submitted records into a single VCV record, which is for variant 1. So this lets you view all the information about this variant together. And that's what you see by default when you look at a variant page in ClinVar is the VCV record. However, you'll notice that 2 of these submitters classified this variant for condition 1, and the other 2 submitters classified this variant for condition 2. Now, these 2 conditions might really be 2 distinct conditions, and so it might be useful for you to know specifically what do we know about this variant condition 1 and what do we know about this variant condition 2. So for that reason, we also aggregate the data by the variant condition pair. So we would aggregate these 2 SCVs into an RCV record that's specific to variant 1, condition 1. And these 2 RCVs would get aggregated into a separate RCV record that's for variant 1 and condition 2. So that's the way we aggregate data into 2 types of aggregate records. So you can see if these are 2 very distinct conditions, you can see why the RCV records are pretty useful, but I'm also going to demonstrate to you why the VCV record is also useful. So here's an example of a variant in KCNQ1. And there's a table on the VCV page called Conditions Germline, which gives you a summary of the diseases for which we have a germline classification in ClinVar. So you can see for this particular variant, it's been classified for several different conditions. You think that'd be pretty useful. You want to know, is the information about Jervell and Lange-Nielsen syndrome different from what we know about short QT? However, when you start to look at the list, you see some complexity. So we have both congenital long QT syndrome and also long QT syndrome 1. So if you already know that you're looking at a variant in KCNQ1, you may not really consider these to be distinct conditions, and maybe you really don't want them to be on separate records. So maybe you want to look at this information together. There's the added complexity that one group is submitted to ClinVar for this whole set of diseases. And this goes back to the case I mentioned earlier, where sometimes a laboratory classifies a variant for a set of diseases because they don't know which one is correct. This laboratory probably doesn't know the patient's phenotype, and so they don't know which disease they even should be considering for this classification. So they have classified the variant for this set of diseases with some uncertainty about which one is correct. In that case, you also don't want to look at classifications for this set of diseases in isolation. You really want to look at all of this information together. And so that's why the VCV record can be helpful to show you all the information within the same context. But then we do provide this table summarizing the information per condition in case it is useful to see information aggregated by the variant and disease. So this row essentially gives you that RCV level aggregation. Disease, the classification for the disease, and the review status. Okay, so that's our segue to the next thing to talk about, which is aggregate classification and review status for VCV and RCV records. So this is the aggregate classification and review status for the VCV record shown right at the top of the page. And then these are the aggregate values for the RCV records shown in this table. So let's talk a little bit about how we calculate those values. I'm going to describe what we're doing for VCVs, but essentially we do the same thing for RCVs. It would just be on the smaller subset of submissions specific to each disease. So again, we're calculating a review status on the VCV record, which gives the user an idea of what level of review has gone into this classifications of this variant and the aggregate. And therefore, it's kind of a proxy for how much should I trust this. So the first thing we look at is we're basing this on all of the submitted data. So the first thing we look at is there a submitted record or an SCV from a practice guideline or an expert panel? And if there is, we use that review status from that submission. So if there is a practice guideline, the variant becomes a 4-star record, and the variant's review status is practice guideline. Or if there's a submission from an expert panel, then the review status for the variant then also becomes reviewed by expert panel, and it's a 3-star variant. And we stop there. Even if there are 50 other submissions for the variant, we don't consider those at all. We just stop at the highest review status for that variant. Okay, but as I mentioned earlier, most variants in ClinVar have not been reviewed by an expert panel, so typically we go to the next question. So we then look at, are there multiple SCVs that have assertion criteria and evidence? So essentially we're asking, are there multiple 1-star SCVs? If there are, we then look at their classifications. So do their classifications agree? If they do, that's pretty high quality. That means we've got multiple submitted records that provided transparency into their classification rules and their evidence. And in doing so, they agree on the classification. There's consensus. So that's a pretty high level of confidence. So that's a 2-star review status. And the review status is actually criteria provided, multiple submitters, no conflict. So that is shorthand for they provided their assertion criteria and their evidence, and there's consensus among multiple submitters. Okay, in the case that their classifications do not agree, that's a slightly less trustworthy situation. So that gets 1 star, and the review status is criteria provided, conflicting classifications. In this case, we have transparency into their process, but we don't have consensus. Okay, as you're probably aware, many variants in ClinVar are rare, so it's often the case that we don't have multiple SCVs with assertion criteria and evidence. So the next question we ask is there a single SCV that provided their assertion criteria and evidence? And if there is, that is also considered a 1-star variant, but in this case, the review status is criteria provided, single submitter. So you can see there's 2 different ways you can be a 1-star variant. They both mean that there's transparency into the assertion criteria and the evidence for that classification, but there's not consensus, either because there's a conflict among multiple submitters or because there's only a single submitter, so you can't have consensus with only 1 submitter. So if there are no 1-star SCVs on that variant, then that means we only have 0-star SCVs left. And in that case, the variant is then also considered 0-star, and the review status is whatever it is on the SCV, either no assertion criteria provided or no classification. So this is a bit of a complicated process, but again, it is documented on our website, so you can review that for more information. So review status is important because we then also use that for calculating the aggregate classification. So again, this is the value you see at the top of a ClinVar record. It is intended to give you an overview of what do we think the classification of this variant is, considering all the information that we have and considering the varying levels of quality of information. What do we think the overall classification of this variant should be considered? So we do this based on the review status of records on that variant, and we only consider records with the highest review status. And so we follow a similar path that we just followed. So first of all, we look and see, does this variant have an SCV from a practice guideline or an expert panel? And if so, the aggregate classification is whatever the practice guideline or the expert panel said. I will note that in the past year or so, we've made a change to this so we can have multiple classifications from practice guidelines or expert panels for different diseases. We don't just have to resolve it to a single value here, because we know we're getting to the point where there's enough expert panels that we may have different classifications for different diseases from different expert panels. So there can't just be one value. So most of our records are not from expert panels. So again, we typically go to the next question which is, are there any 1-star SCVs? And again, we ask if there are conflicts in their classifications. And if there are, the aggregate classification is this term, conflicting classifications of pathogenicity. So you would see this term at the top of the page, and then there would also be an explanation. There'd be a list of what those conflicting values are. So we don't try and resolve the conflict. We just let you know there is a conflict, and then you can work through the data yourself. If there are no conflicts in classifications from the one-star SEVs, we use the classifications on those records. So for example, in the case shown here, the one-star records all said it was uncertain significance. So they were in agreement. If one submitter said it was pathogenic and another said it was likely pathogenic, you would see pathogenic / likely pathogenic here. So not every difference in term is considered a conflict. We have a kind of a slightly complicated set of rules that we use to determine what's a conflict and what value gets reported here. I don't have time to go into all of that today, but it is all documented on the website. And then finally, if we don't have any one-star SEVs, if we only have 0-star SEVs, we do a similar set of questions here. So we ask on 0-star SEVs, are there any conflicts in the classification? Again, if there's a conflict, we just report that there are conflicting classifications of pathogenicity. And if not, we use the classification from the 0-star SEVs in the same way we do here for 1-star. Okay, so now you know a lot about how data comes into ClinVar, what are the major pieces of data that we calculate and how we aggregate data. And so next we're going to shift gears and talk about searching for data in ClinVar. So I have several use cases I'm going to cover today, and I'm going to go from more specific kinds of searches to broader searches. And so we're going to start with searching ClinVar for a specific variant. And my best recommendation is if you're looking for a specific variant is to search using a full HDBS expression. So that's an accession dot version colon with the C dot or G dot portion. So this is a great search term. It's very specific. It typically gives you a single result like the one shown here. I'll note that we do some processing and translation of query terms. And so sometimes you'll see the result of that in the search box here or on the ClinVar website. So often we try and interpret what your search term is and then look for it in one of the indexes that we use for search. So that's what this means. This tells you how we translated your search. So there are a couple of caveats to searching on a full HDBS. One is that it works best if you are searching on the most recent version of a RefSeq. The second issue is that sometimes you don't have the accession.version number for the HDBS expression, particularly if you're looking at their literature. Journals have not always been good about requiring full HDBS. So sometimes you may not have this full piece of information. So there are other approaches you can take as well. So for example, you can just search on the C dot portion. That works. It's just not very specific. So for example, if you search for C dot 19 C to T, you get 400 results. And you can see a lot of these, it's just that there are other genes that have a variant at C dot, that's a C to T. You'll also notice that this second one here, it's actually not C dot 19 C to T, it's C dot 39 C to T. And typically what this means is that there are multiple transcript variants for this gene. And on a different transcript for this gene, this variant is located at position 19. So I will mention this a few times in this section, but whenever you get multiple search results in ClinVar, I encourage you to look closely at those results and make sure which one is exactly the variant that you are looking for. Variant nomenclature can be complicated. Another good way to make your C dot search a little more specific is to add the gene symbol. This is typically a pretty specific search. So in this particular example, you go directly to the page that you want. Again, I will say occasionally you'll run across a gene and a C dot where the gene has multiple transcript variants, and there are multiple transcripts that have a variant called C dot 19 C to T or whatever your C dot is. So again, if you ever do a query like this and you get multiple results back, you want to look at them carefully and see if you can resolve which of them actually corresponds to the variant that you're looking for, because the transcript really does matter. Another newer feature that we've added to search is searching for a specific variant using what I'm calling nomad format. I don't think this is formally a format, but it is the way that Nomad refers to variants on their website. So this is chromosome dash start dash ref dash alt. So you can now use this format. You can use this using dashes or colons. I will say it's a pretty specific search, but not the most specific because it doesn't tell you what assembly you're looking at here. I believe we're defaulting to GRCH38 for this format. But if you want to be specific about it, you can add the assembly onto the end here in parentheses. And then there's a couple more examples here to note that you can, the example I used here was for an SNV, but we can also do this for small insertions and deletions. Okay, next let's look at doing searches for related variants. And the first search I'm bringing up here is using an RS number, which is the identifier from dbSNP. And I'm mentioning this one first because I think often people use an RS number as a way to search for a specific variant, but actually it's not a search for a specific variant. An RS number doesn't represent a specific allele. An RS number represents a type of variation at a location on the genome. So for example, this RS, RS206076, it represents single nucleotide variation at the genomic location corresponding to this position. So you can see this RS represents the T allele, the C allele, and the allele found in this transcript, which is G. So this RS number represents 3 different alleles. So searching on an RS number in ClinVar often does give you a single result just because often it's only the alternate allele that's been reported to ClinVar, but often does work like a specific search. But there are cases like this one where you do get multiple alleles back. So I just want to make sure that you're aware that an RS number can give you back more than one allele. And that can be considered a feature because sometimes you do want to see all of the variants that might happen at this one position. So this can be a good search for looking for several related variants all at the same position. So other kinds of searches you might do to find several related variants would be searching for all the variants that cause a specific protein change. So here I searched for V600E. The second result that I got is the one that you're probably expecting. So this is the SNV and BRAF that causes V600E. But you'll notice there's actually also delins in BRAF that causes the same protein change. There's another gene here, CTNNA3, that has a V600E causing variant. And you'll also see there's 3 variants in BRCA1 where V600E is not the primary protein change, but BRCA1 has many, many transcript variants. So there's probably some other transcript where each of these variants causes a protein change that can be described as V600E. So again, you can add the gene symbol to this to make it a little more specific. That's a good way to go. And I'll also note that I use the single letter protein change here in my query, but you could also use the P dot part of a protein HGVS as well for this kind of search. So we have a couple of more new features looking for related variants. And the first of these is looking for any change at an amino acid position. So let's say you're looking at FBN1 and you want to say, show me all of the variants that cause a change at the cysteine at 1138. So you could search for FBN1 P.cis 1138. You don't specify what the change is, you're just saying any change at this position. And you can see we've got 4 results for that. So we've got variants that cause a change to tyrosine, serine, phenylalanine, and arginine. So this is a nice way to find several different variants that cause a change in the same codon. Again, I'll note I use the P dot format here, but you could also do this with the single letter codes. You could just do C1138. Don't specify what amino acid you're changing it to, and it'll look for any change at that position. And then we have a similar feature that we've added for finding any change at a nucleotide location. So let's say you're looking at MSH2 and you want to see what are all the changes reported to ClinVar at position 942. So you could search for MSH2, C.942, and you can see we have 2 alleles at that location. So we've got G to T reported as well as G to A. And then you'll also notice there's a couple more variants here that are not 942 in the preferred description here. But again, they're probably transcript variants where each of these variants is at the position 942 on that transcript. So again, another encouragement to look carefully when you get multiple results in a ClinVar search. All right, so now going even more broad on your search. Let's say you're interested kind of looking at the protein in general, and maybe you want to look for all changes to an amino acid, but anywhere in the protein you'd find that amino acid. So let's say you want to look at FBN1 and all cysteines in FBN1 that are changed. You could search for FBN1 p.cis, and that gives you about 1,300 results. So this gives you every position in FBN1 where there's a cysteine that has a change that's been reported to ClinVar. So again, a nice broad search if you're doing something a little more different than just classifying a single variant. Even more broad than that, you might be interested in looking at all variants within a gene. So you can search for the gene symbol. You don't have to do the gene square brackets thing here. This is where the search query has translated it for me. I just typed in MSH2, and our search knows that that's a gene symbol. You do need to use HGNC approved nomenclature for this so that we recognize it as a gene symbol. And you may also want to use the variant length filter that's over here on the left side for less than 1 kb for single gene. That lets us show you a nice graphic that's focused in right on this gene to show you all the variants and how they're distributed across the gene. And then the results themselves are the table is shown below that graphic. Okay, and let's go one step broader again. If you're looking for copy number variants or other kinds of large structural variants, you might be looking for genomic regions. And there's a few ways you can do that. The first is what I refer to as UCSC style. So this, again, it's not an official format, I don't think, but it's what UCSC uses on their browser. So it's chromosome number colon start dash stop. Again, this is a pretty good search term. It's not specific to the assembly, and I think we are defaulting here to GRCH37. So if you want to be sure that you get the assembly you want, you can just add it to the front here. So add GRCH37 or 38 with a colon. And when we recognize you're looking for a genomic region like this, we can show you this graphic that shows you the genes in the region in green across the top, a heat map of small variants beneath that, and then all of the large copy number gains or losses below that as these blue and red bars. The table of results is even further down the page, so you can use the graphic or the table to navigate to variants of interest. You can also search for a region using the nomad format. This is similar. It's just the chromosome number dash start dash stop. So this gives you a similar graphic. This has the same issue that it's not specific to the assembly, so you can add that in this format. You add it to the end in parentheses. And then finally, this is just for completeness. This is the format that we use in Entree. Entree is the search system that ClinVar uses. Either of the previous two queries that we do, we're going to translate it to this. So if you ever want to bypass that step and just do exactly what we're doing, you would do it like this. There's a separate field, index field for searching on GRCH38. So I don't imagine that most of you would do that. Most of the time you're probably copying, pasting something from UCSC or Nomad or somewhere else in one of those other formats. But just for completeness, this is the way, the native way to do it in ClinVar, clunky though it is. There's a few other kinds of data that you can search for in ClinVar. You can search for disease or phenotype names. These can be difficult to get exactly right. So you might consider searching on an identifier instead, like a MIM number. You can search on submitter names. Again, you may be hard to get it exactly right. So the page I showed you earlier where you can get to a submitter page and then use the link to get to all of a submitter's records might be a more efficient way to search for all of a submitter's records. You can also search by PubMed ID. If you're ever looking at a paper and you want to see if all of those variants are in ClinVar, you can search for that PubMed ID. These are less common cases, so I'm not going through all the steps to do this today, but there are instructions on the website for doing all of these searches. You probably want to go to our advanced search to do these if you're interested in that. Okay, in the last stretch here, we're going to go through navigating your way through a ClinVar variant page. So when you do a search in ClinVar and you click on a record, this is the page that you go to by default. So it's the VCV page. As you know now, that's aggregating all of the data, all of the submissions for a specific variant, regardless of the disease or other condition it was classified for. So the top section of the VCV page has this blue bar that tells you the variant that you're looking at. And then there's a classification summary. So it gives you the germline classification. And then if there are classifications in the somatic context, you would see the somatic clinical impact here and any oncogenicity here. And also now you know this is the aggregate classification that was calculated by ClinVar based on submissions, and the aggregate review status calculated by ClinVar and also represented by the stars. We have similar aggregate values of the classification and review status for clinical impact and oncogenicity as well when we have submissions. The next section on the page is variant details. This gives you general information about the variant that's true regardless of the classification. So it's things like identifiers, the location on the genome, HGVS expressions. And then that's followed by a table with information about the gene. Most variants in ClinVar are within a single gene, so typically this table just has the one gene with links to the NCBI gene database, OMIM, and ClinGen dosage sensitivity curation results. For copy number variants that span several genes, this table would have multiple rows, one for each gene in that variant. As you scroll down the page, there's then a set of tables that give you information about classifications for the variant in the germline context. So the first of these is the table called conditions germline. We looked at this earlier when we were talking about RCV and VCV aggregation. This gives you a summary of the conditions that were reported in germline classifications of the variant and the RCV level classification and review status. That's followed by a table that has each submission with a germline classification. So this is all of the information that came on each submission. Each row here is a submission. You can see who the submitter is, what their classification was, what's their assertion criteria, what condition did they classify the variant for, and then any additional information like their publications and their comment. These two submissions happen to be from clinical testing laboratories, and they'll typically give more information. We talked about that earlier. But sometimes there are submissions that have more rich data, so I encourage you to use these arrows to expand the rows. So for example, this submission from UDN has really rich data about clinical features present in the individual with this variant, the zygosity, the age, and the sex. So often there's more information to be found by expanding this table. The submissions table is followed by a table about functional evidence. Most records in ClinVar today don't have functional evidence, so you just get this message letting you know that there is no functional data, but if you have it, please consider submitting it. But when there is functional data, it looks something like this. Again, this is another submission from UDN. Submitters can tell us the functional consequence of the variant, the methods that they use to assess the function, a summary of their result, and then a link out to a publication for more information. And this is actually an area that we're working on improving. So look for a nicer display and more data-rich submissions later this year. And then finally, in this section of germline information, there's a table of citations that are related to germline classification of this variant. This table is a great place to start your literature search for a variant. And I do emphasize it's a good place to start. It's not necessarily a comprehensive list. Citations are only included here if they were included in a submission to ClinVar. My team is not curating the literature, finding every paper that's relevant for a variant. So it's important for you to know when you use this table, it has to have been included in a submission to be included here. And when you do your ClinVar submissions, it's a lot of use to the community if you include relevant papers so that they get included in this table. As you continue scrolling down the page, if we have somatic classifications for the variant, we have a similar set of tables for those somatic classifications. So first would be a table called conditions somatic that summarizes the tumor types for which we have somatic classifications. And again, this is the RCV level aggregation that tells you what's the overall clinical impact or oncogenicity for this variant and this type of tumor. That is followed by a table of submissions. For somatic classifications, again, each row is a submitted classification. This table is a little more complicated because there are two types of somatic classifications. The first section provides submissions that are about clinical impact, and then there's a second section of the table with submissions that describe the oncogenicity of the variant. And then finally, after that, there is a table of citations that are relevant for somatic classifications of the variant. Again, these all come from submissions, so it's a great place to start your literature search, but it's not necessarily comprehensive. Finally, at the end of the page, there is this link to TextMine citations. This uses LitVar, which is a tool developed at NCBI by our colleague Xiang Liu. And so it's a way to text mine PubMed Central for papers about relevant variants. The way we use it is using the RS number, which you now know is not specific to an allele. It's for a type of variation at a location. So we do have this warning here that this is a useful feature, but when you get your LitVar results, make sure you know if that paper is about the allele that you're interested in or a different variant at that same location. So that covered a lot of information today. I do want to encourage you to check out the documentation that we have on our website. There's this navigation bar that's accessible from the homepage. And if you check out these menus, a lot of information I talked about today is documented. I need to add a few of our new search features there, but most everything I've talked about today is available on the website. So I encourage you to look around. And I also encourage you to contact us with any questions. You can email us at this address with any question. This goes to the team, to me and my team of curators, several of whom are on the call today. It doesn't go to a black hole. We answer every email that we get. And it's helpful for you to email us because number one, it gives you the answer that you need, but also it helps us understand what's not clear to our users and where do we need to improve things. So please do contact us when you need more information. And then finally, I just want to acknowledge the team that supports ClinVar. On the left is our team of developers, followed by our team of curators who process submissions and answer user mail. So that's Garth, Jenny, Rama, Taiba, and George. Guangfeng is our UI UX expert. And then this is our leadership within the Medical Genetics and Human Variation Program and our leadership within the NCBI and the NLM. So I think we have a few minutes left for questions. Do we?
Oh, we are right on time, but I would say if there's any burning questions, we can definitely address them. But if not, I know the ClinVar team is super responsive to any questions that are emailed through. So if we don't have time for it now, definitely send them through. Not anything in the chat, but we are at time, so we might wrap up there for today. But thank you so much, Melissa, for that hugely informative overview of ClinVar and then some of the newer features as well. This meeting's been recorded, so if anyone wants to revisit it, it will be up online on ClinGen's YouTube channel. So we'll send out the link to that later. But again, thank you so much, Melissa, for that, and thank you to the whole ClinVar team for this wonderful resource.
Fantastic. Thanks, Alicia. Thanks, everybody, for being here today.
Thanks.
Bye-bye.